Алло,
Саламатсыңдар ма?
баба.
Сәлеметсіз.
Сәлеметсіз апай.
Апай даусыңыз ақырын естіліп тұр.
Сөзім естіліп тұр ма?
Сәлеметсіз апай.
Қыздар саламатсыңдар ма? Даусым естіліп тұр ма?
Естіліп тұр апай.
Қалдарыңыз жақсы ма бәрі?
Иә, жақсы бар.
Жақсымыз апай.
Түгел қосылмады ғой, менде 64 адам отыр тек қана. Так, уақыт басталды, бастайық, иә? Сабағымызда бүгін 11-ші лекция, lecture number 11. The title is Genetic Engineering of Bacteria. And the plan of the lesson, the first is the DNA diagnostics, medicines, the production of insulin, therapy of cystic fibrosis, peptide vaccines, and the production of interferons. The development of technologies of genomic sequencing, bioinformatic analysis, and the creating of the recombinant DNA, as well as knowledge about the genetic physiology and the biochemistry of bacteria, are powerful base for the practical application of recombinant microorganisms for practical purposes. And the first is DNA diagnostics. DNA diagnostics is one of the most modern high-tech research methods about organisms contained in genome, and the pathogenesis of the bacteria is determined by the presence of the specific genome.
Anybody do that too before?
Так, қыздар келісіп алайық. Микрофонды өшіре тұрыңыздар. Кім сөйлеп жатқан? Этиканы сақтайық, жарай ма? Let's start. This diagnosis is based on DNA hybridization, and the target DNA is fixed on the membrane. Then a labeled DNA probe is applied to the membrane. Hybridization is performed, and the hybrid molecule is identified by the identifying the label. As the DNA target blood, urine, and the fecal samples are used, washed off from the throat without the preliminary cleaning, contact screening of recombinant DNA with the use of the pathogenic and the non-pathogenic strains. And the next slide. Гүлсезім, слайд көрініп тұр ма сіздерге?
Апай көрініп тұр. Мен жаңа емес көрініп тұр.
Widely used in the diagnosis is the infection disease, allowing you to detect even single microorganisms in the human body. DNA diagnostics combines several research methods, but the most common today is the PCR method. And do you know about this method? And there are more and more test systems on the market that are designed to detect pathogen of the various diseases, mutation of human, animal, and plant genes, and others. PCR analysis is becoming more and more popular in science nucleotide sequences of the genomes and of a number of pathogenic and conditional pathogenic microorganisms have been decoded and the specific pathogen sequences have been identified. And the DNA diagnostics is a replace the developing of field as the use of the PCR and specific probes and significantly increase the sensitivity of the test and allows the use of non-radioactive chromogenic and the chemiluminescent and the fluorescent registration systems. In the many cases, PCR with the subsequent electrophoretic separation of the products is sufficient to detect the mutation of exogenous DNA of an infection agent in the test sample. And with the help of DNA diagnostics, it will be possible to detect the most common genetic and infection diseases and as well as neoplasms. And then next slide about the medicines. Before the advent of the DNA technology, drugs based on human proteins were obtained in the small quantities and their production was very expensive. And with the help of new genomic technology, drugs are obtained in the quantities sufficient of the testing and is used in the clinic. Currently based on the complementary DNA of various human proteins, drugs are obtained for the treatment of various human diseases. The annual volume of the world market of medicine based on human proteins is about $150 billion and is constantly growing. Next slide about insulin and several experimental approaches are used to obtain complementary DNA of target protein genes and clones into vector system. First, it is the use of the tissue saturated with the desired proteins. And, an example of this approach is the production of recombinant human insulin. For the first time, is a complementary DNA was obtained on the basis of the messenger RNA from the pancreatic tissue cells from the which total messenger RNA molecules was isolated, and which was dominated by insulin mRNA. The main protein synthesized by the pancreatic cells is the insulin, and about 70% of the mRNA isolated from the cells in protein. And the mRNA was used to produce insulin complementary DNA, which was cloned into a bacterial vectors to develop a technology for production insulin based on recombinant of the Escherichia coli bacteria. And the next slide about the interference. This approach was not applicable for proteins, the number of which in the cells extremely small and the place of the synthesis is not exactly known. And in this case, the enrichment technique was used. It was first used the production of alpha, beta, and gamma interferons of human, and which have important therapeutic significance, and the complementary DNA of interferons was isolated from human leukocytes, and the first mRNA molecules was obtained from leukocyte cells. The mRNA was fractionated in size and the reverse transcription was performed. And the resulting cDNA was cloned on PBR 322 plasmids in the cells of Escherichia coli. The hormones. The strategy of the construction new proteins by the replication functional dominance or using the direct mutagenesis can be used to enhance also weaken the biological process of protein, and for example, native human growth hormone, HGH, binds to the both the growth hormone receptors and the prolactin receptors in different cell types. To avoid undesirable side effects during treatment, it is necessary to exclude the attachment of the HIH to the prolactin receptors and assign the site of the growth hormone molecule that binds to the receptors in the 8 amino acid sequence and only partially coincides with the site of the molecule that interacts with the prolactin receptors. And it was possible to selectively reduce the binding to the hormone to the later. And the next slide, therapy of cystic fibrosis. An incurable inherited human disease is cystic fibrosis. These patients suffer from the infection disease that affects their lungs. Treatment of recurrent infection with antibiotics leads to the appearance of the resistant strains of pathogenic bacteria. Bacteria and their release products cause the accumulation of viscose mucus in the lungs and making it difficult to breathe. On the offset components of mucus is high moleculars and DNA, which is replaced from the bacterial cells during the release. And the scientists isolated the expressed DNA's gene and enzymes that breaks down high molecular DNA into the shorter fragments. And the purified enzymes is injected as a part of the aerosol in the lungs of the patients with cystic fibrosis. And it cleaves DNA molecules. The viscosity of mucus is reduced, which facilitates breathing and improves the patient's condition. Antibiotics. The production of antibiotics by microorganisms, by the fungi, actinomycetes, and bacteria is a complex process and is determined by the final expression of the biosynthesis and the secretion genes in the medium of the products itself, and as well as the genes responsible for the antibiotic resistance of bacteria to this product, and therefore change in individual gene do not ultimately lead only increase the product yield. And the high productivity is usually shown by strains that have resistance to high concentration of antibiotics. And this property must be taken into account the design and super products. And the recombinant DNA molecular technology has a great effect in this production. Next slide about the vaccines. Recombinant DNA technology makes it possible to create a new generation of vaccines. The introduction of the vaccines causes many formation of the antibodies, and which is a subsequent infection with a pathogenic lead to its inactivation, block proliferation, and prevent the development of the disease. And the type of this vaccine is attenuated vaccine. Modern vaccines are based on the inactivated pathogens or live but non-virulent strains. And the pathogenic microorganisms are modified by the removing the genes responsible for virulence from the genome, while the ability to cause is the immune response is preserved. And the example of such vaccine is the anti-Florella vaccine created on the basis of Vibrio Florella strains from the whose chromosomal DNA molecule the enterotoxin gene is tolerated. Such modified microorganisms are used as live vaccines. Next slide about the peptide vaccines. They produce antibodies in the host body, in the response in the infection, under the presence of the only purified surface proteins is sufficient. Such vaccines contain a fragment of the pathogenic organism. And the recombinant DNA molecular technology is also used for their development. And the advantage of such vaccines is said it does not contain additional antinucleic acid, which can have inserable side effects, and the Sputnik vaccines are developed on the base of the influenza A and B viruses, hepatitis, and the B viruses, rabies viruses, herpes viruses, and the other types of the viruses. An example is the peptide vaccines for the hepatitis B viruses. It is based on the fragment of the protein of the outer shell of the viruses with a size of up to about 200. Next slide about the plant-based vaccines. A revolutionary direction is the development of vaccines based on transgenic plants in the genome of which the corresponding fragment of the genome of a pathogenic microorganisms has been invaded. The data indicate on influenceable perspective in the development and the practical use of such vaccines under this method of immunization of the and the high cost effectiveness of plant-based vaccines is also important, and they give that many experts predict that cost extensive genetically engineered vaccine will increase. And the price of many newly developed vaccines will be higher than the cost of vaccine used in the practical work. The next slide about the preparation of the bacterial restriction enzymes. And more than 3,000 restrictions were allocated, and more than 600 restrictors are available of commercial drugs and used daily in laboratories to modify DNA molecules and solve a wide range of genetic engineering problems. And the restrictors with the various recognition sites as a main tool of genetic research in genetic engineering and the molecular biology. These enzymes are synthesized by the different microorganisms to obtain restrictors their genes and clone using the universal methods. And they have the 5 methods. What first is the bacterial DNA is cleaved by the hind 3 restrictors. Second, the resulting DNA fragments are cloned in the PBR 322 plasmids after its 3 with the hind 3 restrictors. And the recombinant plasmids are transformed into the cells in the molecules of the bacteria Escherichia coli and infected with a lambda phage. If the cells are resistant to the action of the phage, the cell contain restrictors. The cells release and determine the activity of resistase. And the 5 is these positive recombinant clones are used to produce proteins. And this approach is used to isolate genes of various bacterial restriction enzymes having a present of the commercial importance. And obtaining vitamin C. Based on the genetic engineering methods, it's possible to obtain microorganisms with a new properties, for example, with new enzymatic activity, and currently intensive chemical methods are used for the production of vitamins. For example, several chemical and microbiological stages are used to produce vitamin C. The initial raw material is deep glucose from which is a series of the restriction as the last stage. And the next slide about the scheme of the ways of to obtain vitamin C, and it consists of three stages. One of these is a chemical pathway for obtaining vitamin C. Second, the microbiological pathway for obtaining vitamin C, and the genetic engineering technology for vitamin C production. And then the next slide about the microbial insecticides. Insects can cause huge damage to the crop. To combat them, the natural microbial insecticides and they becoming increasingly recognized when they do not have a harmful effect on humans and they quickly destroyed in the environment. And to increase the effectiveness of microbial insecticides, it's possible to manipulate the genes, and in particular with the genes of the infections of the spore-forming soil bacteria, Bacillus thuringiensis, or insect baculoviruses. The insects are safe, highly specific, and effective. The most studied and frequently used insecticides are bacteria of Bacillus thuringiensis. And in the next slide, you can see the list of the reference to explore. And all books, all materials we have in our library sites. And thank you for your attention. If you have questions, please ask. Вопросы, сұрақтарыңыз бар ма? Так, енді группадан шықпаңыздар, мен қазір белгілеп алайын, кімнің бар, кімнің жоқ. Who's absent, who's not absent. Так, группа, сұрақтарыңыз бар ма лекция бойынша түсініксіз жерлері?
Жоқ, апай, белгілеп алдыңыз ба?
Белгілеп алдым, иә. Сіздер СРСП бойынша көп адам маған жіберген жоқ СРСП-ны. Не үшін? Маған бірінші сіресті үшін баға қоюым керек.
Апай, мен жібердім ғой.
Иә, жібергендеріңізге мен қазір қоямын, иә, тексересіз. Ал кім балл жоқ, значит, ол студент маған жіберген жоқ тапсырманы, сіресті айтып.
Апай, мен жіберіп едім, бірақ балл жоқ.
Апай, универге салып қойдық қой біз.
Иә, универге салсаңыздар, қазір қарайын, түгел тексеріп балдарыңызды қоямын. Бірақ кейбіреулері жіберген жоқ. 2 сіреспадан жіберген студенттер бар.
Апай, ол 9 бен 10 шаптан сіреспалар ғой, ә? 11-ді енді жібереміз ғой, ә?
Иә, сіздер маған бір семестрда 2
Хорошо.
ғана балл қойылады. Ал екінші сіреспаға доступ енді ашылды. Енді салсаңыздар болады. Тексерем.
Жарайды.
Сұрақтарыңыз бар ма тағы да?
Жоқ.
Апай, осыдан сіріспесі пятницаға дейін ғой, ә?
Пятницаға дейін емес, бұл апрельдің сіздер 13-ші апта біткенге дейін. Сіздерде 13 апта ғой оқу. 13 апта біткенге.
Апай, сонда мысалы біз 2-2-ден уже 4 сіреспа тіркеп қойық, одан кейін басқа сіреспа керек емес, е?
Бірден 4 сіреспа тіркемейсіздер. Алдымен біріншісін жібердіңіздер.
Иә, білем ғой, 9-ға 1, 10-ға 1 тіркедік.
Иә.
Болды.
Жақсы, сұрақтарыңыз болмаса, қазір сағат 1:05-те кездесеміз, е?
Жақсы, апай.
Жақсы болыңыз.
Сау болыңыз, апай.